discussed, substances can move in and out of cells through diffusion. Remember, diffusion is the movement of substances from an area of high concentration to an area of lower concentration. I've prepared 3 agar cubes so that we can watch diffusion happening. Agar is a jelly-like substance made from an algae. These agar cubes also contain phenolphthalein. Phenolphthalein is an acid-base indicator. What that means is that in an acidic or neutral environment, phenolphthalein will remain colorless. However, when phenolphthalein is exposed to a basic environment, it will turn pink. We are going to put these 3 agar cubes in sodium hydroxide. Sodium hydroxide is a basic substance, and as it diffuses into the agar, they will begin to turn pink. These 3 cubes have 3 different sizes. The small cube is 1 centimeter by 1 centimeter by 1 centimeter, or 1 centimeter cubed. The medium cube is 2 centimeters in each dimension, and the largest cube is 4 centimeters in each dimension. Now, you may notice that these cubes aren't perfectly cut, but that's okay. These measurements are very close to what they are in reality. Before we get started with this experiment, make sure you record a hypothesis on your handout. If you need to go ahead and pause this video to do that, feel free. One more thing, this experiment is going to show us why it's important that cells are small, so keep that in mind as you watch. I'm going to take these 3 agar cubes and place them in the beaker and fill it with sodium hydroxide. From there, we'll wait about 10 minutes to give the sodium hydroxide a chance to diffuse in, and then we'll take a look at our cubes. Okay, now that it's been about 10 minutes, let's take a look at our cubes. So here we have our smallest cube. So what we want to do with our cubes is to cut them in a cross section so that we can see the inside as well. Alright, so I just cut our cube in half, and you can see that the inside has turned completely pink as well. So what that means is our sodium hydroxide has diffused all the way through our smallest cube because the entire thing has turned pink. Alright, and here's our medium-sized cube. So again, I'm going to cut it in half, and we're going to look at the inside. This cube looks different. The pink has not made its way all the way through the cube. The middle remains colorless. Let's take a look at our last cube. So here's our last cube. Again, looks pink on the outside, but let's cut it down the middle. So in the center of this one, we have a very large colorless region. The pink is really only on the outside of the cube. So as you can see, the cube with the smallest volume turned completely pink, while the cube with the largest volume turned pink only on the very outside edges. This is because diffusion happens at the same rate regardless of the size of the object or its volume. Comparing these cubes to cells, if the cells were too large, diffusion would not happen quickly enough to support the cells. For example, cells need to bring nutrients in and get waste out. If the cell were too large, like our large cube, the nutrients would not be able to get in fast enough and wouldn't reach the center of the cell, and the same can be said for the waste leaving the cell. This is why when organisms are complex, instead of having larger cells, they have a larger number of small cells. So hopefully this helped you understand why cells are small and how diffusion works. Go ahead and record the data we collected on your handout and answer the questions.